DNA replication. What is DNA replication and what is its purpose? DNA replication is the biological process in which a single DNA molecule is copied into two identical replications. This occurs in every living organism. The purpose of DNA replication is to have another cell with a duplicated genome. It is a specific phase of the cell cycle that allows cells to actively divide to have the exact same genome. Let's start with an overview of DNA synthesis. There are three basic steps in DNA synthesis. Initiation is the first step where replication begins at a specific sequence of bases on the double helix called the origin of replication. DNA helicase is the enzyme responsible for opening up the double helix. Elongation is the second step where the primer is used as the starting point and new complementary nucleotides are added to the DNA template strand. The synthesis of the new strand can only be formed in the 5' to 3' direction. Termination is the final step where the primer is removed and DNA ligase connects the new complementary DNA strands into continuous strands with phosphodiester linkage bonds to create a complete backbone. Let's go into further detail about each of the enzymes involved in DNA replication. DNA replication begins with initiator proteins. These are proteins that recognize the specific DNA sequence within the origin of replication. DNA helicase is the protein responsible for unwinding the parental DNA double helix into two single strands by breaking the hydrogen bonds between the base pairs. Hydrogen bonds are the intermolecular forces that link the base pairs of the DNA molecule to form the double helix structure. A Y-shaped region is then formed when the DNA double helix is separated into two single strands and copied. This is the replication fork. As this happens, topoisomerase comes into play. This is the protein that relieves the tension caused by the coiling of the DNA helix as it is being unwinded by the DNA helicase. This enzyme cuts the DNA to promote unwinding and rejoins it again. In addition to this process, the single-stranded DNA binding proteins prevent the two DNA template strands from forming a double helical structure again. The next important enzyme is primase. Primase is a type of RNA polymerase and is responsible for synthesizing the primer, which is an RNA strand that starts the complementary strand of the DNA template strand, supplying the 3' OH end for the DNA polymerase to start from. DNA polymerase then is the enzyme that catalyzes the DNA synthesis into the 5' to 3' direction only. Once all of these enzymes complete their respective steps in DNA replication, the leading strand is complete. The leading strand is the new strand of DNA that is synthesized towards the replication fork in the 5' to 3' direction. This is also known as the continuous strand, as the formation of this complementary strand continues on, without breaks, in the same direction how the parental DNA helix splits.
However, you may notice that we come to an issue with the other strand. DNA polymerase can only synthesize DNA in the 5 to 3 direction. But due to the original DNA molecule's anti-parallel structure, one template strand will continuously be replicated towards the replication fork, but the other template strand can only be replicated in the direction opposite of the replication fork. This strand is called the lagging strand. Let's go into further detail about the lagging strand. It is a discontinuous strand where gaps will form as this particular DNA template strand undergoes replication. As mentioned before, lagging strand replication is discontinuous because the complementary strand of the DNA template forms in the opposite direction away from the replication fork. For the lagging strand, DNA synthesis occurs in the same manner as the leading strand up until the point where primers are put down to allow a starting point for the DNA synthesis. Different from the leading strand, complementary fragments are attached to the primers called Okazaki fragments. Okazaki fragments are short, complementary DNA fragments that are synthesized on the lagging strand, each with the starting point of an RNA primer. Okazaki fragments form because of the DNA's anti-parallel nature. Finally, the primers between the Okazaki fragments are removed and replaced with deoxyribonucleotides by DNA polymerase. An enzyme called DNA ligase forms phosphodiester bonds between the fragments to complete a continuous complementary strand.
Because there is a lagging strand, when the final primer of the lagging strand is removed, there is no way for DNA polymerase to synthesize DNA. Because of this, the end of the template strand, called a telomere, It's detected by telomerase, which attaches an RNA template onto the DNA strand until the strand is long enough. Once long enough, the primer attaches to allow a starting point for DNA polymerase to synthesize the end of the complementary strand. Without this, chromosomes will continue to shorten, losing important genetic information. Prokaryotes, on the other hand, do not have this issue of end replication because they have circular chromosomes, so there is no end to the chromosome.